Welcome, everyone, and thank you for joining us. Please note that attendee cameras and microphones will be muted during this activity. Use Zoom's Q&A button to submit questions to our speakers. Our program will begin shortly. Thank you.
Hello, everyone. I would like to welcome every participant to our quarterly CME series on lysosomal disorders. Today's presentation is on new insights into the lysosomal disease from Pathophysiology, which is changing treatment, presented by Dr. Gregory Grabowski. I also would like to thank our sponsors on the behalf of CheckRare and LDRTC, who are the joint participants and the producers of this series. For today's presentation, I would like to introduce the subject with a brief introductory remarks on new insights into the pathophysiology of lysosomal diseases. So these are my disclosures. And to briefly introduce the subject, I would like to start with the personalized or precision medicine, which is attracting a lot of attention recently, and which is taking the therapeutic area for the development of new therapies, especially in cancer. However, a physician or any healthcare provider that takes care of patients with rare disorders, we are very much aware, or we have been very much aware, that N usually equals to 1, at least at the beginning. So all our patients are unique in a sense that how they present with the disorder and how they progress and what their course would be. So I'm just going to start with a quick series that we published about 16, 17 years ago. And the children in that series some of them are adults now. And unfortunately, some of them have succumbed to the disorder. So if you look at this bar graph, these are the normal controls, but all these children have one same disorder called Gaucher disease, which affects the nervous system, or Gaucher disease type 3 or neuropathic Gaucher disease, which is the presentation of deficiency of the lysosomal enzyme glucocerebrosidase. When you look at the enzyme levels, all children had almost not similar enzyme levels, and their phenotypes did not actually represent each other, even though there was a flare of presentation. So if you think linearly, the disease starts with a culprit. In the lysosomal disorders, this is the mutation of a gene that leads mostly to enzymatic deficiency, and the histologic activity starts, and the pathology occurs when you start seeing the signs of symptoms of the disorder. In linear thinking, that the disease starts, the initiation of the process, and then progresses at different speed at different tissues that leads to multiple downstream events. And as the disease takes its chronicity, we start seeing the complications. However, this is a very simple presentation or linear depiction of the disorder. We know that diseases are not linear. They are three-dimensional, or actually more, with the psychosocial effects or the impact of the disorder. So I'm going to show you the 3D genomics. I put the slide as genetics is not what you think, but probably the biology is not what you think. So at the lower panel, you see the chromosomes. This is in the metaphase. They're all spread apart, but in reality, with different techniques that we know now, the chromosomes in one single cell at least, they don't look like that, and they are more like in a clump. So the disease pathology or the process causes the systems to be off from their path, and the system actually impacts the disease process with the genetic and the other environmental sources. So, with this word, I would like to introduce our speaker today, Dr. Gregory Grabowski, who obviously needs no introduction in the disorders in the field of lysosomal disorders. Dr. Grabowski received his MD degree from University of Minnesota, and after his tenure at Mount Sinai for 13 years, he became the chair of human genetics and director of human genetics at Cincinnati Children's Hospital Research Foundation. He's currently the professor emeritus of molecular genetics, biochemistry, and microbiology, as well as pediatrics at the University of Cincinnati College of Medicine. He published over 350 scholarly works on the basic and clinical sciences of lysosomal disorders. He was the founding chair of the American Expert Medical Committee of the Project HOPE, as Genzyme Sanofi Gaucher Initiative. It's a philanthropic program to treat Gaucher disease. And then after his retirement, he worked at multiple pharmaceutical companies as the chief scientific officer. Currently, he's an independent consultant to several companies and foundations that develop therapies for the unmet needs for those disorders. Dr. Grabowski, we thank you for participating in this program, and we're looking for a very interesting talk.
Well, thank you, Ozlem, for introducing me and inviting me to this interesting series of lectures on lysosomal disorders. I'm Greg Grabowski, and I've been in lysosomal disorders for roughly 45 years, and things are changing. And we're going to talk about the pathophysiology and pathobiology of lysosomal disorders and how they're going to impact treatment as we move forward. The learning objectives here, these are my disclosures. I have no relevant financial or non-financial interest to disclose. Now, obviously, since I've been in this field for so many years, what I'm going to talk about is going to be a little bit skewed to what I know and what I have experience in. But the general principles will apply to all of the LSDs in various different ways. But the different degrees and types of cellular and tissue and organ involvement have to be taken into consideration with each of these lysosomal disorders. And it's not just a garbage bag. It's a very heterogeneous, dynamic, and cell and tissue specific metabolic or sensor control organelle. And that's different than what we learned about many years ago and thought things were so simple with the lysosome that it just digested stuff. Well, it does, but it does a lot of other things also. Here's a perspective on LSD pathobiology with a couple of examples. And I'm going to start with the initiation of a disease process by primary or secondary lysosomal storage materials. This, of course, happens in Gaucher disease types 1, 2, and 3. But obviously, since there are 3 types, this is more complicated than just the storage material. In lysosomal acid lipase deficiencies, either the rapidly progressive infantile form or the childhood adult form have differences in their presentation and different kinds of mutations in that gene. Fabry disease, there are several different variants. It's not just the storage of GB3. And there are the mucopolysaccharidoses, especially types 1, 2, 3A, and 3B, that involve many organs of the body and in different ways, but yet seem to store similar kinds of materials. And then we have a group of disorders that are initiated by unknown mechanisms. And these are the ceroid lipofuscinoses or neuronal ceroid lipofuscinoses. We have really no idea of exactly how the pathophysiology in these diseases work. So the historical view for the pathogenesis of a lysosomal storage disease started, of course, with a monogenic abnormality, creating a defective protein, enzyme, or other type of compound in the body. leading to the lysosomal accumulation of undigested or partially digested or degraded substrates, leading to constipated cells and tissues. And of course, the part of this slide that we had missed in the past was not the engorgement of the lysosome, but the inflammation and pain that came along with that. And we'll talk a lot more about that as we move forward. And of course, that led to an idea that the disease severity would have to be based on the severity of the mutation. I dislike that term severity, but it's rampant in the literature, and it is foundation for a thing called the continuum hypothesis, whereby the amount of enzyme that you have determines the type of pathophysiology, and anybody that has a different amount of that residual activity would have the same basic pathophysiology, but would have a different manifestation of it. And I disagree a bit with that. And let me show you why. This is on the left-hand side of the screen, two forms of Gaucher disease type 2. The first form is called the perinatal lethal form of Gaucher disease. And as you can appreciate, the skin is very abnormal. There's a lot of neurological abnormalities as appreciated here by this spasticity and, of course, the flexion of the neck. These children die within a few days to a few weeks after birth. Why? Because they have no glucocerebrosidase activity at all. They cannot make ceramide to provide a permeability barrier of the skin, and they dehydrate and die, as well as having severe neurological finding. Now, with a little more enzyme, you get this phenotype, which is the classic type 2 phenotype at roughly about 5 months of age or a little bit more. Large liver and spleen, obviously cortical thumbs and neurologic involvement with retroflexion of the neck, and of course, strabismus and cachexia. So you could say, yes, this is a continuum from one form to another, and probably a very similar kind of pathophysiology. The same might be said when we look at Gaucher disease type 1. And we're going to show a series of mutations here. The N370S, a very common mutation in the Western world. L4P, a very common mutation in this form of Gaucher disease type 2, but not in this form. These have very complex mutations. A mutation, 84G, which is basically a null. And what we did was we looked at the age of diagnosis and detection in people with these genotypes. And there were a large number of individuals in these studies. And you can see the spread of the data and the ages of onset. But what is very curious is the following. N370S84GG has an age of onset of approximately median age of onset of approximately 5 years. And this is a null. So you have only the N370S. The N370S L4P has a little more enzyme activity. And there you have a median age of onset of 15 years. And then when you're homozygous for N370S, you have an age of onset of approximately, a median age of onset of approximately 30 years. So this is very interesting that it would spread out like this and actually say, yes, indeed, earlier signs and symptoms show up here. And it is true, not only for age at diagnosis, but for the size of the liver and spleen and for their abnormalities, they follow the same type of curve. But of course, there's one other thing going on here too, which is the spread of phenotypes and when they present. It's massive here in the N370S homozygotes. It's less so in the N370S L4P, and even less here, although there's some outliers all the time. The red box that I brought up is a note of caution. These studies were done when we allele-specific categorization of the mutation. Now, of course, the standard is to do whole GBA1 gene sequencing so that you really find everything that's going on. These people here most likely have an N370S and something called a delta 55 mutation, which is a null. So it's like this one. And it was not detected using allele-specific genotyping. So we have to be very careful about when in time and so forth. But this is also a continuum. N370S homozygotes have twice the activity of these people, and N370L44P have not half, but probably maybe another 20% compared to the 84GG mutant. So we have a continuum of the disease, age of onset, and the manifestations. Now we move on to this conundrum of type 3 Gaucher disease. And these are some patients from Poland with type 3 Gaucher disease. And they're all young children, and they have different kinds of manifestations. This little girl, of course, has hepatosplenomegaly and a little bit of neurological findings. This boy is severely involved, although he's not even as old as she is. And he has kyphoscoliosis, a thing we do not understand, as well as hepatosplenomegaly. And here we have two siblings at different ages. He has more neurologic involvement as well as visceral involvement than she does. So how would you explain that with all the same mutation, L44P homozygotes? And it gets worse. These two patients were patients of mine. They are L44P homozygotes. And she was diagnosed at 18 months of age, and she was diagnosed at 8 months of age. And we treated them with enzyme. You could tell they are very happy people, and they have grown up here at 7 and 12 years. They are now 26 and 28 years old and perfectly normal. They have no CNS involvement. Their bone structure is fine, and their hepatomegaly has disappeared completely. But they have L44P homozygosity. So what would explain this? This cannot be explained by residual activity in a continuum of pathophysiologic processes. This is a multiply complex disease. So there is, in certain types of Gaucher disease, there is a continuum within each type, but not between types. And in type 3, it remains a conundrum. Similarly, we see this kind of variation in lysosomal acid lipase deficient patients. Here was a patient reported by Dr. Crocker back in 1965 at about 3 months of age or so. Here's a patient from Dr. Simon Jones, and one from Andrew Burrow, a trainee of mine. Now, this patient is about 4 months old, has massive hepatosplenomegaly, ascites, and liver failure. This patient had a similar kind of phenotype and was treated with enzyme therapy. But now this is not hepatomegaly and splenomegaly. This is mesenteric lymphadenopathy, an unexpected consequence of staying alive with this disease, where you accumulate the cholesterol and the triglycerides in lymph nodes of the mesenteric system. And we're seeing this now too in treated patients with Gaucher disease who have severe abnormalities of their glucocerebrosidase. So these are very different. And of course, the intervention led to shall we say, the finding of this lymphadenopathy. Now, the question is, what do the patients with the later onset variant of lysosomal acid lipase deficiency, so-called cholesterol ester storage disease, what do they look like? They look like you and I. And they have that much variation. There are some younger patients who are significantly ill, but many of them are not. And they have lipid abnormalities that are treated by lipidologists who do not work them up for cholesterol ester storage disease. But in that patient that I just showed you, there's a publication by Porter, just came out. It shows what happens when you give enzyme therapy to the rapidly progressive infantile variant. And without treatment, in historical controls, they die at about 3.5 months of age on average. On enzyme therapy, with replacement of lysosomal acid lipase, they do extremely well, although there are some fatalities because these are extremely sick people. And then there's this mesenteric problem. What do we do there? And what Porter and the group did in England was they treated the patients with enzyme for some period of time and then did a hematopoietic stem cell transplant in conjunction with enzyme therapy and dietary substrate reduction. So it improved, the transplant improved intestinal mucosa, the histopathology, malabsorption, and the mesenteric lymphadenopathy. So a single therapy is not going to treat this disease. And these patients, because of the special way that the brain handles cholesterol, these patients do not get brain disease. The oldest living patient now with the rapidly progressive form has been on enzyme therapy since very early in life, and he's about 15 years of age, and he's an honor student with no neurologic findings whatsoever. So there's this intertwining of initiation and propagation with or without therapy that we really have to explore. And the initiation, of course, is relatively simple at the beginning. You start to collect some storage cells, and the cells can tolerate a certain degree of abnormalities caused by the substrate accumulation, and eventually it starts to fall apart. And that evolution then kicks in cell and tissue-specific involvement of increasing complexity. And there's heterogeneity of organ and region involvement, tissue involvement, and cellular type involvement. And this propagation leads to emergent properties, things we could not predict from the patients, either with or without therapy. And these things can be independent of the initiating event, and eventually become self-propagating. So even if you fix the fundamental problem, you still have a progression of the disease. Now, these principles are all time dependent. And we have lysosomal composition, shape, contents, and numbers different between cell types and the effect of disease expression. And this is very time dependent. It gets worse with time without therapy, and even with some therapies. Cell metabolites differ between organs, tissues, and cells, leading to differential and very specialized involvement of these organs, tissues, and cells. For example, you could ask, why do neuronal ceroid lipofuscinoses involve neurons and almost no other tissue at all? Just those cells. There's a metabolic disruption in a lysosomal storage disease that involves many cellular organelles. The lysosomes are moving around inside cells all the time and attaching and detaching from the plasma membrane, mitochondria, peroxisomes, and they're talking to them all the time, and those organelles are talking back to them. Now, I'm going to show you a little picture here of an animation of the pathophysiology of Gaucher disease, and you can apply this to many others. So we start with stem cells are transferred to monocytes. The monocytes travel to organs through the bloodstream, and they become in those organs macrophages, but the macrophages are all different in the tissue organs. And of course, we have activated macrophages that are eating the materials that they're supposed to digest, and it accumulates, and all of a sudden they start to spew out all these cytokines and cause a pro-inflammatory state that in some patients is very, very severe, and in others is less severe, but is present in essentially all the patients. And the continued accumulation of these cells and their byproducts lead to the tissue manifestations of the disease with bone deformation, enlarged liver and spleen, and so forth. And this is the kind of thing that we have to think about with each of the lysosomal diseases. Now, what are all these other processes that are going on? And I'm going to have you take a time to get to know and love a compound called mTORC1, mechanistic target of rapamycin C1. This is a master controller of metabolism in cells. And it controls the catabolic pathways when it dissociates from the lysosomal membrane. Usually, it's attached to the lysosomal membrane, and when it becomes detached, it can no longer phosphorylate. TFEB and TFE3. What does that do? These master transcription factors turn on autophagy and lysosomal biogenesis. So you create more autophagy. You start to eat yourself more and more. This is like a starvation and stress pattern. And whether it's mitochondria, whether it's peroxisomes, or whether it's just material that in the cells, you start to deliver these to the lysosome and they accumulate more and more lysosomal dysfunction, decreased protein function, leading to altered integrity of storage materials, and substrates are increased, precursors are decreased, and there can be direct activation of a whole pro-inflammatory pathway, including inflammasome activation by interleukin 18, by RIPK1 and RIPK3 can lead to necroptosis. Here, the pro-inflammation can lead to apoptosis or pyroptosis. So very complicated pathway for just a lysosomal disease, but it gets more complicated because this is only applicable to many parenchymal and non-immune cells. When you look at myeloid cells, for example, macrophages and neutrophils and some other immune cells, you see the following. Let's say in a storage disease, mTORC-C1 continues to be bound to the lysosomal membrane. It's not totally dissociated. What happens then is it can phosphorylate some of the TFEB and TFE3, slowing down the autophagy, lysosomal biogenesis, and all the other effects, and increasing the anabolic pathways. Catabolic is decreased somewhat, anabolic pathway is increased, leading to increased fatty acid oxidation by the mitochondria and by the peroxisomes, increasing the energy pool so that you get endolysosomal expansion. More lysosomes, more parts of the cell occupied by lysosomes. These are for antigen processing and cell migration, and the cell structure and function is remodeled by this pathway. In addition, TFEB and TFE3, the parts that are not phosphorylated, are functional and kick in cytokine and chemokine production, leading to a pro-inflammatory state. So it's cell-specific and disease-specific in when these kick in. I use this simple diagram of an inflammatory hepatic disease model in the lysosomal acid lipase storage diseases. Now, the hepatocyte is generally the basic target organ, target cell of the LAL deficiency. And that accumulates cholesterol esters and triglycerides in microvesicular steatosis. This creates a lipotoxicity and hepatocyte damage. Then you have macrophages that are derived from the bone marrow who have much larger vacuoles filled with material that enter the liver and create more of a pro-inflammatory pool of cytokines and chemokines. And you have another type of macrophage, which are the Kupffer cells that are derived from fetal liver macrophages. And they have a different type of pro-activation and pro-inflammatory system. This leads to a fibrotic environment in which the hepatic stellate cell produces collagen, leading to fibrosis and eventually cirrhosis. So you have two types of cells involved here, mainly, which are inflammatory cells and hepatocyte. But the inflammatory cells are from different ontogenies. And that's important because they have different impacts on the pro-inflammatory system. And this is true for all of the lysosomal diseases, not just in the liver, but in other specialized tissues. So this is a beginning understanding of some of the autoinflammatory and autoimmune disease that can occur in the lysosomal diseases. This is kind of a complex, wordy slide, but let's go through it. There's involvement of the innate immune system in all lysosomal storage diseases. But the nature and type of that immune response is unique to the type of LSD. So let's start by those that store heparan sulfate. There's inflammation in the brain, the CNS, and in bone via the TLR4 pathway. So the toll-like receptor 4 pathway. Both the CNS and visceral diseases are affected in all mucopolysaccharide diseases except mucopolysaccharidosis type 4 and 6. The brain is spared. Why that is not totally clear. What is clear is if you use TNF-alpha antagonists, you decrease the inflammation, it improves the CNS and skeletal system. So by direct intervention into the innate immune system with these antagonists, you can improve the disease without enzyme therapy. In disruptions of lipid degradation, you get inflammation of many organs. The gangliosidosis show prominent pathogenic neuroinflammation in the CNS and visceral pathobiology in only some. For example, GM1 gangliosidosis and in GM2 Sandhoff's disease. There are neutral lipidoses. They can be predominantly, like in Gaucher and Fabry, or exclusively visceral, like in lysosomal acid lipase deficiency, with different kinds of storage materials. Or they can involve both the visceral organs and the CNS, as in the neuropathic forms of Gaucher disease, or the CNS and the PNS in Krabbe disease. So complicated. And, for example, the immunopathology for Krabbe disease is not well worked out right now. In the neuronal ceroid lipofuscinoses, you get lysosomal dysfunction, but the pathogenic mechanisms are largely unknown. They're lumped together as their ceroid lipofuscinoses because that's the nature of their histologic involvement. But you can actually have abnormalities in the neurons of the brain in model systems before you have storage of the lipofuscin. It's isolated brain disease in only certain parts of the brain. It's neurodegenerative. And the question is, why is the specificity? There's neuroinflammation present, but it may not be the main driver of the neurodegeneration. We don't know what is. TPP1, which is in CLN3, leads to neuroinflammation and something very interesting, DAMs, disease-associated microglia. And these were first found in Alzheimer's disease models. And they are very characteristic transcriptome patterns. And they're being found now in more and more diseases, including Gaucher disease and some of the mucopolysaccharidoses. And there may be targets here for repurposing some drugs that are being made to dampen their activity in neurodegenerative disease that are more common. Maybe there's a chance to use some of that in lysosomal diseases. CLN11 is very interesting. It is a progranulin deficiency, and this is a molecule that has 8 different subunits to it. So it's much like prosampicin, which has only 4, but this is 8 different subunits to it. And homozygosity for progranulin deficiency leads to an early onset form of neuronal pseudolipofuscinosis. The pathophysiology is poorly understood, as is the biology of progranulin. And interestingly, the people who have heterozygosity for mutation in the progranulin gene, GRN, develop frontotemporal dementia. They're predisposed to it. They don't all get it. It's partially penetrant. So this is a very interesting observation because it's similar to what we see with the propensity of heterozygotes for Gaucher disease having a propensity to develop Parkinson's disease. So in mucolipidosis 4, this is caused by defects in a nonspecific channel in multiple organelles. It is a modulator of innate immunity. It is a modulator of TFEB, so it controls the whole system. And it has the ability to clear substrates from cells by exocytosis. There are a series of 400 genes that TFEB interacts with that cause exocytosis of lysosomes and therefore putting some of the substrates out into the circulation. And this has some pluses and minuses, but it gets rid of the storage product from inside the cells. Part of this immune response that we have in our cells and that the lysosomal disease individuals have was dissected out by unbiased lipidomics. And these people took cells and did all the lipids in the cells and clustered them statistically, like you see here in this circular pattern. These are co-regulated lipids in different parts of this circle. And so these are co-regulated, all these are co-regulated. These are regulated in a different manner, but they're co-regulated and so forth. And what they found was on the plasma membrane, if you had the lipidomics there, from the pattern that you see of the co-regulated lipids, you could predict the nature of the TLR response, in particular the TLR4 response. And it differed in Krabbe disease, Gaucher disease, Farber disease, and this disorder of lysosomal stability. So this is very important that this changes the nature in which cells respond in an immunologic manner. Now, this is an example of the CLEAR system, and it means coordinated lysosomal expression and regulation. And the question about this clearance from the cell by activating this gene from ML4, you would then export substrates out of cells. And the question is, would you break tolerance to these substrates? This could lead to the activation of the adaptive immune system with antibody formations and reactions. So the question becomes, since lysosomal diseases are autoinflammatory diseases, are they also autoimmune? And we found in Gaucher, as an example, that it is indeed both. Certainly, the macrophages are highly activated, spewing out chemokines and cytokines in an untreated state, but they also spew out the stored material, glucosylceramide or GL1. Somehow tolerance is broken, and the body makes antibodies to GL1. Eventually, the plasma cells make a series of these antibodies. You get antibody-antigen complexes, so these are antibodies bound to GL1. They activate the complement system, which then goes back and attaches to its receptor, C5AR1, either on dendritic cells, leading to inflammatory responses, or on macrophages, leading to additional inflammatory responses, and an unexpected finding, which was an increase in translational control, an upregulation of glucoceramide synthase. So these antibodies eventually led to the generation of more GL1 and made a vicious cycle of propagation of this disease. Many of the lysosomal storage diseases have had detected in the plasma antibodies to their substrates. And the question becomes, is the autoimmune system also involved in all of the lysosomal diseases in addition to the autoinflammatory system? So both the innate and the adaptive immune system. Very important finding for potential interventions. So some observations at this point were the accumulated and excreted substrates in the lysosomal storage diseases lead to autoantibody development. In this case, it was glucoceramide, glucosphingosine, or indeed certain types of heparan sulfate isomers. And it's only specific forms of that lead to antibody formation, autoantibody formation. And lysosomal disease models that are pharmacologic or genetic in targeting of autoinflammatory agents have a positive therapeutic effect on visceral bone and CNS. So there are agents out there that could be used. And the mechanistic details of the autoinflammatory and autoimmune LSDs should provide alternative or adjunctive therapeutic targets to block propagators of these diseases. But there's more to understand so that we really get to full understanding of how to treat these diseases. This is an example. This is in situ mass spectroscopy of glucoceramide being looked at in the brain of a mouse on the left-hand side with a Gaucher disease phenotype. What we have here are different types of glucoceramide. This one has an 18-carbon chain, fatty acid acyl chain. Here it's 20, here 22, and here 24. The yellow indicates that it's highly accumulating. The darker colors mean it's lesser accumulating. And you can see how regional, like the C18 is, only certain areas of the brain have C20 is in a different area. Very heavy accumulation of the C22 and posterior accumulation in the C24. And this is only the beginning. This doesn't look at the cell types that are involved because there's differential cellular involvement in addition to the regional. And this is true now, if you think about it, for all of these lysosomal diseases that involve the brain. There are regions of specificity that they cause disease abnormalities. So this is a picture of the macrophage system, the microglial system in the brain. And this was done by work by these people, a spectacular piece of work, and characterizing, for example, the lysosomal content, the properties of the membranes, the unique transcriptomes, of the shape of them in these different areas of the brain. And again, like I said, they migrate from the yolk sac in the embryo to an area and then differentiate and are taught by the developing neurons in this area what they should do, what kind of properties they should have. It's a very interesting developing science. And it means that our therapy has to be based on what that science tells us. This is not a very good overlay, but it helps you get an idea. This is the brain of a mouse that has a neurologic form of Gaucher disease. All the little brown spots are staining for microglia. It's staining with CD68. There are better things to stain with. But what it shows you is that it's not uniform. It's very concentrated in particular areas and sparse in other areas, but it's there. There's a lot of involvement. And in the areas that there is more involvement, you'd expect to have more disease and more disease manifestations. Do we get our therapies to those places? And that's will be very important as we move forward. Now, that's all talking about what happens in the brain, and we talk about autoinflammation and autoimmunity. But what about a connection between the two things? A connection between what is going on in the viscera and what is going on in the brain. And of course, we have the genetic mutation that leads to dysmetabolism in the lysosome and in cells and inflammatory responses, leading to lysosomal disruption and some of the things that we've talked about, leading to something called priming. And what this is like is the anamnestic reaction that you have in the adaptive immune system. Once you're exposed to a pathogen, you form antibodies to it and you have memory cells. And they're waiting and ready to respond to that specific antigen again. Priming happens in microglial cells in the brain. It's not as specialized as it is in the adaptive system. You just have a system set up that is ready to respond in a hyperactive way. So here you have a disease process in the brain that leads to activating signals here with activation and of the microglial cells. There are calming signals also, but they start to have commitments to the different types of things that they can do, the microglial cells can do in the brain. So they're primed and ready to go. What about when you have systemic inflammation? We all know that you can't use enzyme therapy to get across the blood-brain barrier. It doesn't work. The blood-brain barrier, unless it's a very severe involvement with a porous blood-brain barrier, you're not going to get your enzyme across the brain, into the brain. But systemic inflammation is by chemokines and cytokines which penetrate right through the blood-brain barrier. And they can activate the primed cells in the brain, leading to a significant decrease in calming signals and an even greater increase in activation. But what does this do? This is known by adult neurologists for sure. Let's say you have a person with Alzheimer's disease who has cognitive impairment, but they also have gout. And during an inflammatory reaction in their gout, their cognition falls way off. It's not because they have an infection in the brain. It's because the chemokines and cytokines from the gout reaction penetrate into the brain and activate the abnormalities in their microglial cells. Once you cure or fix the gout, their cognition comes back. Now, that's acute. If that inflammation is chronic and continues to depress the brain, it becomes permanent. So we don't know how this all works exactly in the lysosomal storage diseases, but you can tell that some of the patients with, let's say, type 3 Gaucher disease who get enzyme replacement have improved neurologic function once their liver and spleens are shrunk and their cytokine storm is abated a bit. And then when they come off of enzyme therapy and that storm starts up again, they start to have more neurologic problems. Now, those connections have to be looked at very carefully and provide, again, opportunities for treatment. So in conclusion, the understanding of lysosomal autophagy functions continues to evolve. Remaining incompletely detailed temporal, spatial, molecular, biochemical, and immunologic processes of lysosomal storage disease initiation and propagation are still there. We need to know this. We need to work this out. We have tools today that we never had 35, 40 years ago. There's a lack of appreciation for each individual's disease stages at a tissue and cellular level as a necessary prelude to better treatments. For example, we can all look at an X-ray and see a certain kind of fracture on a bone, and we can assess how difficult it might be for that to heal and so forth. But we have no way to really assess, let's say, what's happening in the liver or what's happening in the lungs by taking an X-ray or measuring a biomarker in serum. We need those things to improve our therapy and our targeting. There are inadequately defined, untreated disease natural history. And what I mean by that is now that we're treating all these diseases, the actual natural history of untreated disease has disappeared. And we need people who have a wealth of information about patients who are untreated to be able to share that so that we can use that as a background. Like, for example, in lysosomal acid lipase deficiency, we know that the median age of death is 3.5 months without therapy. And you have only a few patients that live beyond a year and they deteriorate. So this is really important. They're not well characterized. We have inadequately defined untreated disease history as well as phenotypic changes revealed by treatments, like, for example, mesenteric lymphadenopathy is occurring. And then even at this understanding, stage of understanding monotherapy, whether it's enzyme therapy, substrate reduction therapy, or gene therapy, you must have that. But the totality of the disease process, for example, with inflammation, required intervention to improve outcomes. And better outcomes result from early intervention, while the disease processes are less complex and potentially reversible. I would say from our stage right now, we know that the era of monotherapy is fundamentally dead. We have to start broadening our perspectives on how we treat these patients with multiple different drugs. I just want to thank my collaborators for over the last 4 decades or so who did a lot of this work in genetics, immunology. Hong Du and I developed the lysosomal acid lipase deficient mouse and its therapy. Pathology, you need pathologists, and the pathology today is much, much better than it was 25, 30 years ago. We have better tools. And if you do any kind of crystallography or other kind of fundamental structural work, you need somebody as a crystallographer. Thank you very much.
Thank you, Dr. Grabowski, for this very detailed and very intriguing lecture. We have questions in the Q&A panel. I would like to start with the first one. As you described the neuronopathic involvement, even with the same mutation, and I showed the slide. So what the audience is asking, why do some Gaucher patients develop the severe neurological involvement, whereas the others, even in the same families?
Yeah, it's a very important question, and we don't know the answer. One of the best examples of this, of course, is the Norboten form of Gaucher disease type 3. which all derived from a first cousin union in 1654 and is L4P homozygote. And the variation in neurologic involvement can be just like a type 2, or as Lars Fennerholm told me, the oldest patient that he had that developed the first signs of neurologic disease was 56 years. So there's a spectrum, even within a relatively homogeneous population in Sweden, of this happening. So we don't know why that happens, but there are obviously other genes that are impacting the expression of the Gaucher disease type 3. We looked at glucoceramide synthase for variants in glucoceramide synthase and really didn't find much at a genetic level. So I'm not sure that that's the causation, but there could be other things, the turnover of cells, the whole variety, but we don't really have a good idea as to what that's all about, unfortunately.
So I'm moving from the neurological system to the other lysosomal storage disorders that affect not mainly the neurolytic system, but such as Pompe disease as a muscle disorder, not the infantile Pompe disease. So could you comment some of the mechanisms that may be different in a muscle disorder as opposed to Gaucher disease that mainly presents as a hematological disorder?
Yeah, I think that Dr. Rabin's group at the NIH has done an awful lot of work on the autophagy pathway in Pompe disease. And it is significantly disrupted. And it turns out, of course, that the muscle itself is the primary organ that is involved in Pompe disease and is not an immune system cell type, although you do get storage in some of the myeloid cells. So as far as I'm aware, there's not much known about inflammatory reaction in Pompe disease. You could theorize there should be in some cases, but I'm not aware of much of the literature that would point in that direction.
So we're talking about Pompe disease. Let's move to the infantile Pompe disease that we know that there is neurological involvement, especially since the infants are surviving right now. So how can you comment on the continuum of the phenotypes in different LSDs?
Yeah, I think that the reason I have difficulties with the continuum hypothesis is that it really skews your thought process to thinking that all these things happen because of substrate and the amount of substrate. And what you're seeing in Pompe disease is that these individuals, the infantile form, are living longer. They're starting to develop manifestations that we never saw before. But the early pathology, even in infantile Pompe, that was published in the late 50s and early 60s, looking at the brain, there was storage in those brains of glycogen. Now that they're living longer, they're starting to have more disease manifestations, cognitive defects, and so forth, and brainstem findings, which cause some of the breathing difficulties that they have. So I think we have to really look at each individual disease and say, for example, in Pompe, how much of what is happening to patients now has to do with an inflammatory reaction, or is it maybe more substrate? In some of the others, there's more inflammation in addition. But in all of the diseases, you have to treat the primary abnormality, sure. But that's not the only thing you probably have to treat.
Thank you. So I'm going to move forward with the neurological spectrum or the neurological associations with lysosomal disorders, obviously Parkinson's disease or Parkinsonism. has been a very hot topic, and that is now actually being more inclusive in all the neurodegenerative disorders, including Alzheimer and cognitive changes like Lewy body dementia. So, one audience is asking whether Parkinson's disease or other neurodegenerative disorders are late onset lysosomal diseases.
I think this is the crux of the matter when we talk about the continuum hypothesis. We have very little data available from, well, again, we'll pick on Gaucher disease and the heterozygote state in Gaucher disease and Parkinson because it's been the most well studied. But there are over 50 or so lysosomal genes now that have been associated with neurodegenerative disorders. But what happens here is if you think that the Parkinson disease in a Gaucher heterozygote results from substrate, you should be able to see that. And there are no concrete and very solid data that actually show that there is significant storage of glucoceramide or glucosphingosine in the brains of people with Parkinson's disease with the heterozygosity for GB1. What is seen is starting to be looked at now is the abnormalities of the lysosome in some of the cells, particularly some of the striatal neural, the dopaminergic nerve cells, where the attachments of lysosomes to the mitochondrial membrane and the tethering is affected in a very significant way by small changes in glucoceramide in the lysosome or in the lysosomal membrane. So these may be very, very subtle findings, and in many of the disorders that would be heterozygote in the population and possibly affecting these neurodegenerative diseases. So these are going to be very subtle. The other piece that is a challenge here is when you go in and you do look at substrate accumulation, let's say in the substantia nigra, many times this has not been conducted with really good histopathology to tell you what kinds of cells are there, because the lipid content is naturally different in certain cells like microglial cells than it is in neurons. And so the pathology is going to be, histopathology is really going to be important in interpreting what we're looking at if we see substrate changes. And that would apply for any of the lysosomal disorders. And there's a lot of, I think at NYU, Dr. Nixon has been a big proponent of the idea that Alzheimer's disease has its pathogenesis beginning in the lysosome, or at least propagating that disease. So lysosomes are becoming a very important part of neurodegeneration.
Okay, so let's talk about actually the treatment as we started with the pathophysiology. So now the new generation ERTs that are being developed theoretically may access CNS. So how can you comment on this? It is when we start, when is it going to matter when we start the treatment, or how are we going to measure the impact on CNS in a peripheral or closed system?
Yeah, there are two parts of this, and the first is in the talk that I gave, I said multiple times, you have to treat the initial cause of the disease some way, whether it is you give enzyme into circulation and it gets across the blood-brain barrier, or you get enzyme or gene therapy directly into the brain. You have to treat the cause. That's one thing. Then you have to start asking the question, what is reversible? How do you stage the place where you're at with the disease process? For example, what would be the outcome of treating a patient with type 2 Gaucher disease with significant neurologic manifestations, or even mucopolysaccharidosis at a stage in which they're already very severely involved, what would be the expected outcome? Is it stability? Is it reversal? What would you say? And the point is, you have to start early, before all these other mechanisms kick in and start to kill off neurons, for example, and they don't come back. There are some possibilities of replacing neurons, but those are very much into the future, and whether they're going to be specific or not. So, two things: treat early and correct the primary defect, and then we have to stage these patients, develop better ways of staging these patients for seeing how well we could expect a response to occur in any individual. It's complicated.
Very complicated. And I think the more we understand, the more we know that we are very limited in our knowledge. So one last actually question, and probably this is going to be the closure one. So you talked about extensively with immune pathways, the inflammatory components. So how would you incorporate a treatment or management plan by addressing these pathways?
Well, I think there are some good animal data that support the idea that you can intervene in the inflammatory pathways and actually have a very positive result in mucopolysaccharidosis, for example, on both the brain function and the skeletal system. And I think that we need to have more work in that area, and then move those to see if we can repurpose some of these anti-inflammatories that are already approved to lysosomal diseases and that have characterized known inflammatory or pro-inflammatory states. So I think that's what we have to do. Now, when you use that, for example, I can envision having an individual with a very severe early onset form of, let's say, lysosomal acid lipase deficiency, that you treat them initially with, let's say, some form of enzyme replacement or gene therapy or whatever, at the same time that you use that there's a known abnormal pro-inflammatory state, and you pick an anti-inflammatory to treat them at the same time to get them through these very severe times that they're involved. The other part that we can envision some of these potentially anti-inflammatory compounds being useful is in some of the patients with bone disease or even maybe lung disease, that you can treat them on a chronic basis to try and alleviate those problems, maybe even prevent it from happening. Which those are, that all has to be done in animals first with good models. Some of the models for lysosomal diseases in animals are very good, and some are useful, but not real good mimics.
Yeah, that is correct. And the other thing is, obviously, the different lysosomal disorders have different immunobiology, including the tissues that they may affect, such as the MPS on cartilage, Gaucher disease on bone, and whether it's developmental or immunology. We thank Dr. Grabowski again, and with the comments that I have, this was a really wonderful presentation and a learning experience for everyone. This presentation will be available on the web after this, and then please stay tuned for our third CME event later in fall, October or November. We thank again to our supporters, including Takeda, Cheisi, Ultragenyx Pharmaceuticals, and Spark Therapeutics for sponsoring this event. And please, the participants who actually took notes and attended today's participation, please claim the CME credit on ldrtc.cds.pesc gce.com. Thank you very much. Have a great afternoon, and looking forward to see you in the next CME presentation.